Don't necessarily cross out what you have and write what I wrote just because it's not identical. Check to see, is what you're saying, what I'm saying, mean the same thing. Biology, unlike, let's say, math, where when the answer is 5, the answer is 5, there's only one way to write it, is a little bit different because we're using words to explain our answers more often than we're using data or figures. And so sometimes we could both be saying the same thing, but using a different word to say it. So we'll start with question number 1. Why do we need more producers than consumers? I heard a lot of people's answers to this. Does someone want to share what they said there? Why don't you tell me what you wrote because we talked about it. If the consumers were trying to consume more than what is produced, well, they can't. You would need to have more producers. And so she's touching on the idea that because the consumer is eating the producer, there has to be lots of them so that we don't run out. And so I'm going to write not running out as a point. And that's a point that we'll talk about a lot when we get on the topic of food chains and food webs, and that'll become in one of the later units. I'm wondering if anyone had a photosynthesis cellular respiration related answer that they put here. Maybe something about glucose or oxygen or something along those lines. So I'm going to ask you actually to think about what you wrote for number 2, and then we'll come back to number 1. Number 2 says, when does cellular respiration happen and in which cells? And actually the very first few sentences that I said about photosynthesis and cell respiration are when I answered this question for you the first time. Does anyone want to tell me when cellular respiration happens? During the day? Does it happen at night? Does it only happen in the summer? When does it happen? Anytime. So cellular respiration doesn't just happen at night. And a lot of people have that misconception because photosynthesis requires light. But cell respiration can happen anytime, day, night, winter, fall, spring, summer, anytime. Does anybody want to tell me which kind of cells can do cellular respiration? So put up your hand if an animal cell can do cellular respiration, and you say yes to that. Now put up your other hand if a plant cell can also do cellular respiration. I'm putting up both of my hands. You might want to copy me. Both of them can do it. So, plant and animal. So I come back to my first question. Why do we need more producers than consumers? I just said a plant is doing cell respiration. That must mean that plants use some of the oxygen and glucose they produce. They're not giving it all away. If a plant didn't perform cellular respiration, it would not be able to grow and to function. So plants are not giving away every single bit of glucose and oxygen they make. They are using some of it. And so that is why we definitely need more producers than consumers if we're explaining it based on photosynthesis and cell respiration. But the idea of how much energy we can get and how much energy we have is going to come back in other chapters. Now when we talk about the internal parts of the chloroplast, I'm going to draw a picture. The space that's just the fluid is called the stroma. The stack of discs is called the grana, and each individual disc is called a thylakoid membrane. Those are vocabulary words that you will need to be able to use and to identify visually and by their description. If we're talking about the mitochondrion, it's an oval as well, and it has that wavy line on the inside. The wavy line is the cristae, and the inside is the matrix. You need to be able to identify those because we're going to talk about what happens in each of those spots. Now, number 5 is a question, where there's lots of correct answers, I would imagine. I think that most people are good at finding differences between two things. So when I talk about photosynthesis and cellular respiration, a lot of people find it easy to find a difference between them. For example, photosynthesis makes oxygen. Cellular respiration uses oxygen. Photosynthesis makes glucose. Cellular respiration uses glucose. If we talked about the products and the reactants, we see that they are opposites. So that's definitely something that's different between photosynthesis and cell respiration. We could also talk about energy and whether or not it's used or produced. In photosynthesis, I need energy from the sun. In cell respiration, I produce energy at the end. Does anyone have something else that's different between photosynthesis and cell respiration that they'd like to share? Those aren't the only things in the world. Those are just what I think are the obvious ones from what I wrote the other day. You could even put photosynthesis is only in plants, whereas cell respiration is in all living cells. That would be another thing that's a difference. Did anyone come up with a similarity, something that is similar between those two sort of opposite-looking processes? Nobody wants to share? I know some people wrote an answer to this. So if we talk about what's similar between them, obviously we can say we have the same for substances. So even though they're not in the same order, it's the same for things. And so I like to really make sure that we point that out, because if you know the reaction for one of them, you write it backwards and you basically have the reaction for the other. Now down at the bottom, ATP was the last question. And the first point asks you, how does ATP provide energy for vital functions? And it's the energy in the third phosphate bond. So I showed you a picture of ATP, and I said the key thing to note was that there were 3 phosphate groups attached to it. And what happens is that that third phosphate group can come off. And when it does, energy is released that the cell can use to do other things. A vital function would be something that a cell needs to do to survive that it requires ATP for. So I gave you a few examples in your notes, but for example, muscle contraction. Any kind of cell reproduction, active transport, all of those things require ATP. Now the process that creates ATP, well, I could just refer back to my answer to A and say, I have a molecule that has 2 phosphates, and I add a third phosphate on. And the energy for doing that comes from the glucose and oxygen I got from photosynthesis. Now that sums up basically what we talked about last time. Does anyone have any questions that they'd like to ask about that? So then here's what I would like to do today. I would like to talk about a couple of new things in terms of processes and things that happen. So we're still looking at things kind of at a general level. We're going to talk about 2 processes that happen in both photosynthesis and cellular respiration. And then after that, we're going to start talking very specifically about photosynthesis. So if you could please take out your notes. We left off on this slide last time. What is ATP? So what we're going to focus on, and this is from what we talked about last time, is how this phosphate group gets put back on the molecule. Again, you don't have to know the structure of the molecule to be able to draw it out, but you have to recognize that there are 3 phosphate groups and that I can take one off and put it back on. So what we're going to look at is, how do I put that 3rd phosphate on? So this process is called chemiosmosis, and that is a vocabulary word that you need to know. Now, chemiosmosis is the name of the process that helps us put the 3rd phosphate back onto our molecule. So I'm going to read the words that are up here, and then I'm going to explain to you what's going on in this particular picture. Now, I know that some people like to draw pictures for themselves, and some people like to just look at pictures. If you are interested in having a picture of chemiosmosis, and you don't want to necessarily copy that one down, keep in mind that your textbook has lots of really good illustrations. And the page, oh, I'm too far ahead. The page that we would want to look at for that illustration is figure 5.12, which is on page 175. So that's just another reference for an illustration that looks slightly different than this one, but it's giving you the same basic idea. Now, here's what's going on. You have a structure called ATP synthase. That's this molecule down here, this purple thing. ATP synthase is a special structure in the thylakoid membrane. So just to get everybody situated here, when we draw a membrane and we're looking at it from up close, you'll notice a series of circles with little lines, and then the same thing but opposite on the other side. Does anybody remember what that is or what it's made out of? You would have done this in Science 10. Perhaps that was a long time ago. So in the membrane of a cell, you have phosphates and you have lipids. And so that is a cell membrane. Anytime you see a picture that looks like that, with little heads with little legs coming down from them, that's a phospholipid bilayer. And you'll see pictures with that type of illustration fairly frequently in this particular unit, because most of what we're talking about happens on or around a membrane. Now, membrane function is something that's more of a Science 10 topic than a Bio 20 topic, but I just wanted to get you situated. So this structure is in the thylakoid membrane. So what you're looking at in this picture is a really blown up view of one of those little round discs that's inside a chloroplast. That's what you're seeing here. Now here's what ATP synthase does. It provides the only pathway for hydrogen ions to move down their concentration gradient. Now, I asked you to read the other day at the beginning of this chapter some information about diffusion and osmosis. And just so everybody remembers, diffusion is when molecules like to move from an area where they are very concentrated towards an area where they are not. When I say move down their concentration gradient, what I mean is exactly that. Move from an area of high concentration to an area of low concentration. Now, normally, molecules would just do that. But in this case, the membrane is in their way. If they could, they would go directly through the membrane, but they have a charge, and so it's hard for them. So ATP synthase is the pathway. It is the way that a hydrogen ion is going to get from a spot where it's very concentrated to a place where it's less concentrated. So the idea is that a hydrogen ion can only go through ATP synthase. It can't get out of the thylakoid membrane any other way. Now the nice thing about ATP synthase is that it has a little mechanism on the inside. Now it's actually a little bit more complicated, but what I always tell people to think of is that there's a switch. And as hydrogen goes through, it flips the switch. And so once the hydrogen goes through, it helps make ATP by joining a phosphate group and ADP. So there's a phosphate, there's an ADP. As hydrogen moves through, it activates this mechanism, and those two molecules are put together. So hydrogen is trying to get from this internal space out here, and just so we're clear, this is the stroma. So hydrogen ions are trying to get from inside the thylakoid membrane out to the stroma. And then what we'll see later on, up here, there's a pump. And we're actually going to put them back in. And this whole cycle is what helps the reactions that happen in photosynthesis and cell respiration occur. Now, there are other structures in this picture that I don't want to mention specifically right now because we haven't gotten to those reactions. But just so you know, these two chunks here are where the light is being absorbed for photosynthesis. And so that's why there's little green dots on them in my picture. This is chemiosmosis. Would anybody like to ask any questions about this process? So again, it's the structure in the membrane on the bottom that interests us. It's the way hydrogen moves out of the space between the thylakoid membranes to the stroma. And as it does, ATP is formed. Now the other process, and it's really a pair of processes that we have to discuss, is oxidation and reduction. Now this is not the only place that you would run into oxidation and reduction. Oxidation and reduction is like a whole unit in Chem 30, so we are just going to touch on the surface of it here. Oxidation and reduction are two processes or reactions that occur to help break down glucose. And so when we're talking about breaking glucose or making glucose, we have to talk about oxidation and reduction. So, what is oxidation? When we look at it in terms of what's happening in photosynthesis and cellular respiration, we can talk about one specific type of atom or molecule. If we talk about the processes kind of more in general, anytime I say hydrogen, what I really mean are electrons. In the examples that we'll see, it's hydrogen, that's what we're talking about, but lots of molecules have electrons in them. It's just that in our case, hydrogen is the one that we're using. So oxidation is the removal, or maybe let's even say the loss of hydrogen or just simply electrons from a molecule. In our case, hydrogen is going to get picked up by coenzymes. So we already know what a coenzyme is. It's something that makes an enzyme's job easier and faster. Reduction then is the opposite. Reduction is the addition of hydrogen, or we could just say electrons if we wanted to be more general, to another molecule. In our case, where are we going to get them from? We are going to get them from those same coenzymes that picked them up. So what I'm trying to show you is that this is like a cycle. I can't have one of these processes without the other. If something is adding hydrogen, well, something else had to have lost it. If something is losing hydrogen, well, some other molecule has to gain it then. So over here, there's a little picture of a lion and a little thing that says Leo says Gur. And I still remember when I was in chem in high school, like 8 million years ago, I remember our chem teacher teaching us this, and I still use it. It's a little saying that says Leo says Gur, or Leo the lion says Gur. LEO represents loss of electrons is oxidation, LEO. Gur, gain of electrons, is reduction. Now, I've heard other ways for people to remember which one is which. Another one that I find really useful is the saying oil rig. In oil rig, we don't even mention electrons or hydrogen or anything. We simply mean oxidation is loss, reduction is gain. So those would be two fairly simple ways that you could use to remember what oxidation is and what reduction is. So far, would anybody like to ask a question about what oxidation is or what reduction is? So this picture that's on the screen is very similar to figure 5.7 on page 167 of your book. And it's just trying to illustrate to you that I can't have one without the other. If something is going to lose electrons, well, something else has to gain them. If something is going to gain electrons, well, something else has to lose them. The reason I'm showing this picture is actually because I wanted to talk about just two vocabulary words. And the words are oxidized and reduced. Those would be adjectives that I use to describe a molecule after one of these processes has happened. If something has been oxidized, it lost electrons or hydrogens. If something has been reduced, well, it has already gained hydrogens or electrons. And I'm pointing those words out because very frequently I might ask you something about, could I oxidize something, or has it already been oxidized? So past tense versus future tense. Now the other thing I'd like to mention is the coenzymes that are going to do the transporting here. NAD is one of them. You can refer to it as NAD all the time. I wrote down the name there in case anyone is interested. NAD stands for nicotinamide adenine dinucleotide. I won't ask you that, I promise. You can just refer to it as NAD. This is a coenzyme that picks up hydrogen atoms. Now I say here that it's analogous to an empty dump truck, because that's more or less what it is. It is waiting to pick something up. It has not yet been reduced. So NAD, just so that we're clear, can be reduced. It hasn't yet. Once it picks up a hydrogen atom, it becomes NADH, and that's the reduced form of it. So if I was to ask you a question about oxidizing and reducing things, and I asked you, hey, can I oxidize NAD? You would say, no, I cannot. It doesn't have a hydrogen to lose. If I asked you, could I oxidize NADH? You would say, yes, it has a hydrogen that it can lose. Now if I asked you, can I reduce NAD? You would say yes. There's a space for a hydrogen. But can I reduce NADH? No, it's already reduced. And so that little bit of terminology is something I wanted you to see. Now, FAD is the other one. It's called flavinadenine dinucleotide. You can just call it FAD. It is another coenzyme that does basically the same thing. When it picks up hydrogen, it becomes FADH, and there's actually a little 2 there. Sometimes I just write FADH because I don't like for the 2 to be the thing that messes you up. But if we're going to be correct, the molecule is FADH2. Once again, FAD is the oxidized version, and FADH2 is the reduced version. Now the last thing it says at the bottom is that reduction always follows oxidation. And for some reason I can't seem to see the whole thing, so we'll just scooch it over here. With the use of enzymes and coenzymes. The other thing that I wanted to mention about oxidation and reduction is that molecules that are reduced tend to be the ones that have more energy. So, this molecule and this molecule, and any molecule that has already been reduced, is generally something that has high energy. And I'm mentioning that now, and we'll come back to it again later, because knowing what type of molecule has more energy in it really comes down to knowing, is this the oxidized or the reduced form of that molecule? Now, in chemistry in general, you got to think about electrons, but for us, for photosynthesis and cell respiration, look for the H. Does it have H? It's been reduced. If it doesn't have H, hasn't been reduced yet, and that can be your distinguishing factor. Would anybody like to ask any questions about oxidation and reduction? Yes? Generally, for us here, if there's no hydrogen already, I could put a hydrogen on. So we're only going to talk about NAD and FAD. So if I write them without the H, then I could put a hydrogen on. If we're talking more generally, we would have to get into a bigger discussion about chemical structure and bonds and double bonds, which I would be happy to show you later, but I'd prefer not to do now. Okay. Does anybody else have any questions that they would like to ask about this idea? All right.